In vitro hematological effects and attenuation of ultrasound waves in human blood. Abstract in vitro hematological effects of ultrasound have been reported in many studies to understand the absorption and attenuation mechanisms of ultrasonic waves in the human blood. The objective of this study is to assess the in vitro hematological effect and attenuation coefficient of the ultrasound in human blood at different frequencies. Pathological parameters of complete blood count, CBC, tests such as hemoglobin, erythrocytes, leucocytes, and thrombocyte cells were counted after the exposure of ultrasound. Blood samples of different groups, A+, A-, B+, B-, AB+, AB, O+, O, were collected from 23 healthy subjects having mean age, 34.56 plus or minus 14.20, years from BVH, Bahawalpur of Pakistan. Informed consent from subjects was taken to study the CBC test at 370C. The attenuation of ultrasound in blood was measured using Linear Transducer Probe-0805AT for 15 minutes. One-way analysis of variance, ANOVA, test was used to assess the variability of blood cells in samples. A p-value, p equals 0.05, was deemed to be statistically significant in data. In this study, the attenuation coefficient, DOT, of ultrasound waves was measured at frequencies, 3.5 to 11 MHz, using the power law. Statistical analysis revealed that in vitro exposure of ultrasound in human blood exposed a reduced amount of blood cells in CBC test of ultrasound samples, US, as compared to CBC test of control samples, CS. Keywords, CBC, ultrasound attenuation, human blood, frequencies, in vitro. Introduction. Ultrasonography is a non-invasive diagnostic modality which is commonly used in the intracranial radiotherapy. In dental practice, it has been executed for cleaning the tooth and to kill the germs. It has been practiced in urology to remove kidney stones, to see blood flow irregularities and the position of the fetus in gynecology 1. In engineering sciences, ultrasonic testers are commonly applied to detect the porosity, flaws, uniformities, dimensional measurements and the characterization of materials 2 to 3. Furthermore, ultrasound waves, USW, are employed in industrial processes such as plastic welding, material cleaning, and widely used in sonochemistry for 1U, François Th. and Guy Cloutier. 2007, Experimental ultrasound characterization of red blood cell aggregation using the structure factor size estimator. The Journal of the Acoustical Society of America 122.1, 645 to 656. doi.org web link. 2 Samakrutave, Shivaldikan v. Bobro v. Kozlov v. 2006. Development of acoustic methods and production of modern digital devices and technologies for ultrasonic non-destructive testing. Ultragar SA. Ultrasound. 61, 412-21. 3 Samakrutave, 2003, Modern ultrasonic instruments for the non-destructive inspection of metal products. Metallurgist. 47, 7324-9. 4 Phoenix, Christopher Peter ETAL, 2014, High-Intensity Focused Ultrasound Technology, Its Scope and Applications in Therapy and Drug Delivery. Journal of Pharmacy and Pharmaceutical Sciences 17.1, 136-153 doi.org web link. 5 Calorfio MM, Machado J.C. 2006, Measurement of the ultrasonic attenuation coefficient of human blood plasma during clotting in the frequency range of 8 to 22 MHz ultrasound in medicine and biology. 32, 71055-64. 6 Jimenez N., Redondo J., Sanchez Morcillo v., Camarena F., 2015, Nonlinear ultrasound simulations including complex frequency-dependent attenuation. Physics Prostia. 1, 63:108-13. 7 O'Brien JRWD, 2007, Ultrasound, Biophysics Mechanisms. Progress in Biophysics and Molecular Biology. 1, 93, 123-212-55. 8 Amin, VR, 1989. Ultrasonic Attenuation Estimation for Tissue Characterization. 9 Lyons, Mark E., and Kevin J. Parker. 1988 Absorption and Attenuation in Soft Tissues. 2 Experimental Results. IEEE Transactions on Ultrasonics, Ferroelectrics, and Frequency Control 35.4. 511-521. 10 Mazzilli F., Lafon C., D. Holland C., 2014, a 10.5 cm ultrasound link for deep implanted medical devices. IEEE Transactions on Biomedical Circuits and Systems. 8, 5738-50. 11 Skevan, BAA, ETAL, 2009, Therapeutic Ultrasound for Dental Tissue Repair. Medical Hypotheses 73.4, 591-593 doi.org web link. 12 Bradley E., Treeby, ETAL Measurement of the ultrasound attenuation and dispersion in whole human blood and its components from 0 to 70 MHz. Ultrasound in Medicine and Biology 37.2, 2011 289-300 doi.org web link. 13 Shung, Kopping K., Rubens A. Sigelman, and John M. Reed. Scattering of Ultrasound by Blood. IEEE Transactions on Biomedical Engineering 6, 1976 460-467. doi, 10.1109/TBME.1976.324604. 14 Zeinen, PV Theoretical Analysis of Sound Attenuation Mechanisms in Blood and in Erythrocyte Suspensions. Ultrasonics 30.1, 1992-2634.doi.org web link. 15 Zodi T.I., Krona R., 2018, Estimates for the Acoustical Stimulation and Heating of Multiphase Biot Issue. Biomechanics and Modeling in Mechanobiology. 1, 17, 3717-25. 16 Kamimura Ha, Flamant J., Villette J., Caffarelli A., Aaron Boden R., 2019, Feedback Control of Microbubble Cavitation for Ultrasound-Mediated Blood, Brain Barrier Disruption in Non-Human Primates Under Magnetic Resonance Guidance. Journal of Cerebral Blood Flow and Metabolism. 39, 7/11/91-203. Ultrasonic waves have been influenced by the mechanism of scattering and absorption when transmitted through the biologic tissues. 5. Resultantly, the energy and intensity of ultrasonic waves lose due to the heat and molecular exchange of energy in tissues. 6. The mechanism of scattering and absorption lead to classical attenuation coefficient of sound waves that depend on the frequency. 7. When the sound waves pass through a medium, i.e. air, soft tissues, cartilages, bones and blood, its amplitude and intensity are changed as an exponential function of penetration depth or distance. This reduction of amplitude and intensity is known as attenuation. The attenuation of US waves becomes larger in hard tissues and cartilages as compared to soft tissues. 8. However, the temperature effect, cavitation streaming and free radicals effects have been reported in the previous literature. 9 to 10. Ultrasound absorption has been quantified directly by measuring the amount of acoustic energy converted into thermal energy or total acoustic attenuation. 11. Published literature has demonstrated that the ultrasound attenuation in blood shows nominal changes below 10 MHz frequencies due to its scattering of sound waves. Normally, the frequency of US waves is used from 1 MHz to 20 MHz in the medical field. 12 to 13. The scattering of ultrasound waves in human blood depends on the frequency, approximately very close to Rayleigh scattering below 30 MHz, and changes sharply at higher frequencies. 14. Previous studies have demonstrated that the attenuation of ultrasound in the blood depends upon the frequency, absorption mechanism, and the concentration of blood cells. 15, 16, 17. Limited work has been explored about the attenuation of sound waves in the human blood. In this study, we have made an effort to assess the in vitro ultrasound exposure in blood cells such as hemoglobin, erythrocytes, leucocytes, thrombocytes, and the attenuation coefficient in blood at different frequencies. 17 Arshad M.J., Sadiq A.M., Zahid M., Niyama A., Fayaz H., Buzdar S.A., Naeem Ma, Imran M., 2019, Synthesis, Characterization, and Blood-Based Toxic Effects of Superparamagnetic Nanoparticles. Materials Science. 1, 25, 4359-64. 18 Calorfio Marcos M., M., Machado J.C. 2006, Measurement of the ultrasonic attenuation coefficient of human blood plasma during clotting in the frequency range of 8 to 22 MHz ultrasound in medicine and biology. 32, 71055-64. 19 Krishna, G. Gopala, and A. Ahmed, 1990. Absorption of ultrasound in human blood. Le Journal de Physique Colloques 51.c2, c2-311doi.org web link. 20 Mirpur M., Shakerizada A., Basir P., Jamay B., 2016, Effects of Low-Intensity Continuous Ultrasound on Hematological Parameters of Rats. Journal of Biomedical Physics and Engineering. 6, 3195. 21 Salim Sup, M.S., ETAL, 2011 A new measurement method of separation percentage for human blood plasma based on ultrasound attenuation. International Journal of Physical Sciences 6.306891-6898, doi, 10.5897/IJPS 11.1454. 22 Amin, Viren R. Ultrasonic Attenuation Estimation for Tissue Characterization. 1989. 23 Hendy, W.R., and Rittenauer, E.R., 2003. Medical Imaging Physics. John Wiley & Sons. 24 Carstensen L., Schwann H.P., 1959, Absorption of Sound Arising from the Presence of Intact Cells in Blood. The Journal of the Acoustical Society of America. 31, 2185-9. 25 Bamber J.C. 1986, Attenuation and Absorption. Physical Principles of Medical Ultrasonics. 2:93-166. 26 Rico L.G., Junca J., Ward M.D., Bradford J.A., Bardna J., Peterize J., 2018, Acoustophoretic Orientation of Red Blood Cells for Diagnosis of Red Cell Health and Pathology. Scientific Reports. 24, 8, 115705. 27 Johnston, R.L., Goss, S.A., Maynard, V., Brady, J.K., Frizzell, L.A., O'Brien J.R., W.D., and Dunn, F., 1979. Elements of Tissue Characterization. Ultrasonic Tissue Characterization 2, 19-27. 28 Sabo, Thomas L. Diagnostic Ultrasound Imaging, Inside Out. Academic Press, 2004 and Duck, F.A. Physical Properties of Tissue, a Comprehensive Reference Book, London/San Diego, Academic, 1990, doi.org web link. 29 Smith D., Franco R., Dicer C.A., Schneider D.K., McGill C., Smith Q.D., Meyer G.D., 2018, Red Blood Cell Response to Blast Levels of Force Importations into Freely Movable Fluid Surfaces Inside a Closed Container. Frontiers in Physics. 6:93. 30 Williams A.R., Sykes S.M., O'Brien J.R.W., 1977, Ultrasonic Exposure Modifies Platelet Morphology and Function in Vitro. Ultrasound in Medicine and Biology. 1, 2, 4311-7. 31 Treby B., Cox B.T., 2010, Modeling Power Law Absorption and Dispersion for Acoustic Propagation Using the Fractional Laplacian. J. Acust SOCAM, 127:2741, 2748. doi.org web link. 32 Lunan KT, Wen AC, Barfod ET, Edmonds PD, Pratta. 1978, decreased aggregation of mouse platelets after in vivo exposure to ultrasound. Thrombosis and hemostasis. 40, 01568-70. 33 Mirpur M, Shakerizada A, Basir P, Jameh B, 2016, Effects of Low-Intensity Continuous Ultrasound on Hematological Parameters of Rats. Journal of Biomedical Physics and Engineering. 6, 3195. 34 Kismawan IG, Sutapaji N, Ulira IM. 2018, Treatment of 40 kHz continuous ultrasound towards blood cells of mice, Mus musculus L. International Journal of Life Sciences. 19, 2, 366-74. 36 Oleshkevich, AA, 2017. Various in vitro effects of continuous and modulated ultrasound on blood cells of different animal species. Biophysics, 62, 4, 603-615. Materials and methods. To assess the ultrasound properties, 5 blood samples of each group, A plus VE, AVE, B plus VE, BVE. AB plus VE, ABVE, O plus VE, OVE, were collected from 23 healthy subjects having mean age with standard deviation, 34.56 plus or minus 14.20, years in which gender was 15 male and 8 female from Bahawalpur Victoria Hospital of Pakistan. For this purpose, evacuated tube of polypropylene, 3 ml, containing K3-EDTA, ethylenediamine tetrahydric acid tripotassium, size 13 mm x 75 mm and diameter 10.8 mm, Becton, Dickinson and Company, Franklin Lakes, New Jersey, USA, was used to perform the complete blood count, CBC, test at 370C. The complete blood count test was performed in the pathological lab under normal condition. In this study, ultrasound exposure was done in collected blood samples of each group for attenuation purposes. Pathological parameters of blood such as hemoglobin, erythrocytes, leucocytes, and thrombocytes were considered to see the ultrasonic effects. Ultrasound waves were passed through K3-EDTA tube for 15 minutes using Linear Transducer Probe-0805AT, 3.5 to 11 MHz, of Toshiba Nemio XGI style. Blood samples were exposed by ultrasonic waves at different frequencies, 3.5 MHz, 5 MHz, 6 MHz, 7 MHz, 9 MHz, and 11 MHz. The pathological indicators I.E. hemoglobin, erythrocytes, leucocytes, thrombocytes, mean corpuscular volume, MCV, hematocrit, HCT, mean platelet volume, MPV, mean corpuscular hemoglobin, MCH. Lymphocytes percentage, LYM%, in each blood sample were assessed by CBC reports as mentioned in Table 1. However, in this study we focused on blood cells such as hemoglobin, erythrocytes, leukocytes, hematocrit, and thrombocytes to see mean count of blood cells after the exposure of ultrasound. The CBC reports of control samples were compared with CBC of ultrasound samples, US. Statistical analysis was performed using software, Origin Pro.8, www.originlab.com. One-way analysis of variance, ANOVA, test was used to assess the variability of pathological parameters of blood samples. A p-value, p equals 0.05, was deemed to be statistically significant in samples. In this work, power law was used to assess the attenuation coefficient, DOT, as this power law was referred in many studies by Bradley et al., 2010, Marcos M. et al., 2006, Shung et al., 1976, Gopalaji.k et al., 1990, and Salim Miz et al., 2011, to measure the attenuation coefficient as mentioned in equation 1 equals 118. Theoretically, in the case of blood, the power of frequency was taken, n equals 1, so it was taken linear to frequency and can be calculated using equation 2 equals 0.571219 to 20. The attenuation coefficient, dot, of this experimental was measured using values mentioned in table 2. The speed of ultrasound and density of human blood was calculated using equation 3 and 4 adding HCT, %, as mentioned in appendix. C equals 0.9x HCT, % plus 1545 3 equals 0.78x HCT, % plus 1017 412. The comparative study of attenuation coefficient, dot, of ultrasound of this in vitro study was displayed by figure 2. Results. CBC test of blood samples was performed to assess the in vitro hematological effects of ultrasound and the attenuation coefficient in blood cells. In this experiment, 5 samples of each blood group were studied at different frequencies, 3.5 MHz, 5 MHz, 6 MHz, 7 MHz, 9 MHz, and 11 MHz. CBC test of each blood sample was averaged before and after applying US waves. The effect of ultrasound waves in the human blood at different frequencies was mentioned in Table 1. One sample t-test induced a significant difference, p equals 0.001, when hemoglobin, erythrocytes, leucocytes, and thrombocytes of blood samples were compared with and without ultrasound at different frequencies. A less amount of WBCs was seen as compared to normal profile of CBC test. The platelets near about 14.4 to 20% decreased than normal profile and the hematocrit, HCT, i.e. percentage of RBCs in blood was also decreased 21. Hemoglobin and mean corpuscular volume, average volume of RBCs, decreased and packed cell volume was also seen decreased. The number of platelets was also found about 5.5 to 10% decreased as compared to normal profile. One-way analysis of variance, ANOVA, test was used to assess the inconsistency of pathological parameters such as RBC, WBC, hemoglobin, of blood cells. Mean values with standard deviation of control sample CBC, CS, and CBC of ultrasound, US, at different frequencies, megahertz, were displayed in Fig 1. A p-value found, p0.05, remains significant in CBC, CS, and CBC, US, samples. Furthermore, the attenuation coefficient, dot, was measured using power law formula as mentioned in text references 12, 18, and 29. The attenuation coefficient, dot, of blood samples was calculated in vitro at different frequencies. Elementary parameters such as 0.1 equals density of blood, C1 equals speed of ultrasound in blood, C2 equals speed of ultrasound in polypropylene, Z1 equals impedance in first media I.E blood, Z2 equals impedance in second media i.e. polypropylene, X equals K3 EDDA tube diameter, A equals attenuation coefficient at 1 MHz, were taken to calculate the attenuation coefficient as shown in Table 2 and Table 3. Figure 2 displayed the attenuation coefficient using power law by prominent authors. In this study the results found a higher attenuation coefficient at frequencies, 3.5 MHz to 11 MHz, as compared to previous studies. This higher attenuation coefficient trend may be due to K3 EDDA tube material structure in which the absorption, attenuation, relaxation time, and the thermal mechanism of ultrasound vary as depicted in Figure 2. F. Frequency, megahertz, megahertz, CBC, CS, complete blood count, control sample, CBC, US, complete blood count, ultrasound samples, FL equals femtoliter MCV, mean corpuscular hemoglobin, HCT, hematocrit, PLT, platelet count, MPV, mean platelet volume, MCH, mean corpuscular hemoglobin, MCHC, mean corpuscular hemoglobin concentration, LIM, lymphocytes percentage, PWD, platelet distribution width, Pg equals picogram g/dl equals gram per deciliter. Point 1 equals density of blood, C1 equals speed of US in blood, C2 equals speed of US in polypropylene, Z1 equals impedance in first media I.E blood, Z2 equals impedance in second media I.E polypropylene, X equals K3 EDDA tube diameter, equals attenuation coefficient at 1 MHz. Figure 1, in vitro ultrasound exposure in blood, CS, control sample CBC without ultrasound, mean values with standard deviation. Discussion. Human blood assumed to be consisting of blood cells 45% in volume and suspended in plasma under normal hematology analysis. Blood cells were made up of red blood cells, erythrocytes, platelets, thrombocytes, and white blood cells, leucocytes. The CBC test might be varied slightly owing to temperature effect under normal conditions. Previous studies had been used the Power law to assess the attenuation coefficient, A, in human blood at different frequencies as mentioned in references 12, 18, 24, 29, and 30. It was well reported that attenuation mechanism in human blood depends on many factors at the cellular level owing to structure of molecule, intermolecular forces, viscosity, and the thermal molecular properties. 21. Krishna et al. Presented the acoustic impedance of hemoglobin, plasma, and erythrocytes and reported higher acoustic impedance in erythrocytes as compared to whole blood at 3 MHz. In this study, the attenuation coefficient of blood samples was very close to Krishna et al. Work 19. The absorption in the human blood depends upon the viscosity due to a change in density of red blood cells and the surrounding plasma. 24. This mechanism leads to reduce the cell size of blood cells by increasing the tonicity of suspension and the osmosis process across the cell membrane. The scattering mechanism in the human blood was strongly related to red blood cells, including shear rate and the concentration of the plasma protein. That is why the concentration of blood protein was considered a biomarker of scattering and the absorption process at higher frequency of ultrasound. This study has explained the attenuation coefficient of red blood cell at different frequencies. The red blood cell lost its concentration due to application of ultrasound waves and due to the viscosity of relative motion at frequencies lower than 15 MHz. 25, 26. Therefore the less concentration of red blood cells was assessed after exposure of ultrasound as compared to the control samples. In this study we reported the hematocrit versus speed of sound and the density of human blood. The referred density of human blood was taken in 1060 kg/m3, but density equals 1044 kg/m3 was used to assess the absorption impedance, Z, and the attenuation coefficient, dot, at different frequencies. Several experimental studies demonstrated that the attenuation coefficient changes due to the hemoglobin oxygenation as well as temperature. 27-28-29. Schneider et al., 1969, explained that a 4% increase in the attenuation coefficient at 10 MHz in deoxygenated hemoglobin solution as compared with oxygenated hemoglobin solutions. Similarly, the attenuation coefficient decreased 8% at higher frequencies, for example 500 MHz. However, Shung and Wright et al., 1977, elucidated an increase of 60% attenuation coefficient at 10 MHz. In this study the attenuation coefficient of hemoglobin, G/dl, was increased at the rate of 1% at 3 MHz to 11 MHz. It was reported that the attenuation coefficient of ultrasound waves depends upon the concentration of red blood cells as well as blood proteins. This minimal increase in hemoglobin might be ignored at physiologically level 12. However, the attenuation coefficient of hemoglobin rose directly at higher frequencies as shown in Table -2 and Figure 1. Williams et al., 1983, studied the interaction of ultrasound with platelets and found the adverse effect of ultrasound effects on platelet structure. 30. Another aspect of this study was to find decreased counts of platelets under ultrasound exposure. Tribibi et al., 2010, observed the damaged morphology of platelet during exposure of ultrasound therapy 31. Similarly, Lonanke.d et al. also found reduced platelets during the application of ultrasound at different frequencies 32. Bahawalpur et al., 2016, studied the bioeffect of low-intensity effects in the blood in vivo and found reduced number of platelets, hemoglobin, and HCT as explained by this report 33. Ultrasound exposure on the leukocyte cells indicated a minimal effect approximate less than 1%. Kazmawan et al. Studied the ultrasound effect on leukocytes and erythrocytes in blood cell of mice and reported a decline of both blood cells 34. Oleshkevich et al., 2017, investigated the effects of continuous and modulated ultrasound in blood cells of different animal species and reported the leukopenia, cytolysis, destruction, the aggregation of cells, foaming of the cytoplasm of granulocytes, rupture of the cytoplasmic membrane, bursting of nuclei, and their deformation and disturbance of their borders 36. In this study, the leukocyte cell counts reduced in very small amount due to its morphology or high immune susceptibility of cell structure. This in vitro study has made an effort to assess the hematological outcome. Furthermore, the attenuation coefficient, at higher frequencies in human blood might be helpful to understand the physical aspects of ultrasound in medical sciences as well as physical sciences. In this study, ultrasound exposure was not conducted more than 11 MHz frequency. Ultrasound waves at higher frequencies may present noticeable change in blood cells concentration than low frequencies. The limitation of the study was a small data set of blood samples and gender-based interpretation of ultrasound. Conclusion: in vitro study of ultrasound exposure in human blood presented that hemoglobin, erythrocytes, leukocytes, and thrombocyte cells count dropped than normal profile of CBC. The reduced blood cell during the ultrasound exposure was the outcomes of several biophysical mechanisms, for example shear rate, temperature, and frequency of ultrasound waves. This study also indicated that the attenuation coefficient of ultrasound vary at different frequencies, 3.5 MHz to 11 MHz, in the human blood. Shear power law manipulation in previous and current study is not enough to explain the exposure of ultrasound waves in blood cells. Further improvements are requested to estimate the concentration of blood cells during the execution of ultrasound. Therefore, this work may assist to biophysicists to evaluate the ultrasound exposure in human blood as well as to comprehend the attenuation coefficient of ultrasound at different frequencies. Acknowledgement. We are thankful to Professor Dr. Mustangsar Warayach who helped us to perform ultrasound experiments in BVH Hospital in Bahawalpur. Conflict of interest. It is certified that there is no conflict of interest among authors. Author contribution. Muhammad Arshad Javid proposed the idea of the study. Muhammad Imran conducted the experiments under supervision of Dr. M. Arshad Javid. Dr. Quratul Rashid provided us blood samples, contributed to data collection and sampling.